Professor Sarah Gilbert and Dr. Catherine Green. Vaxxers, the inside story of the Oxford-AstraZeneca vaccine and the race against the virus. Narrated by Karen Cass and Ariane Stanley.
In late 2019, news began spreading about a novel coronavirus rapidly infecting people. It would come to be called the SARS-CoV-2 virus, and the disease it caused, COVID-19, became one of the greatest global health crises since the 1918 flu pandemic. By early 2020, researchers at the University of Oxford had started developing a safe and effective vaccine against the virus, and they eventually released the Oxford-AstraZeneca COVID-19 vaccine in record time. Two scientists heading the research team, Dr. Catherine Green and Professor Sarah Gilbert, were there every step of the way. In these blinks, you'll discover how they sped up a process that normally takes years and what scientists and governments around the world can learn from this experience in order to combat the next deadly virus.
Blink 1 of 7.
Co-author Kath Green was staying at a campsite in northwestern Wales when she and her friend struck up conversation with a fellow camper. When Kath lamented the lack of cell phone signal, the camper expressed scepticism about the new 5G towers being installed around the UK, even though no associated health risks have been found. At least, Kath's friend joked, this person wasn't saying that 5G caused COVID-19, or that Bill Gates was inserting microchips into everyone through vaccines. The camper replied that actually, while there might not be a conspiracy, she didn't know what went into these vaccines, and she didn't trust the people making them. She called these people them. Little did she know that them meant Kath. She knew exactly what was in one of the soon-to-be-approved vaccines. In fact, she was part of the research lab at the University of Oxford that had developed it.
The key message here is, scientists were researching viral outbreaks and developing vaccines for years before COVID-19 was ever identified.
To people like that camper in Wales, it might seem like the COVID-19 vaccines were created too quickly and under mysterious circumstances. But the truth is that scientists were already preparing for something like COVID years before the first recorded case. The official name of the virus is SARS-CoV-2, and it's neither the first coronavirus nor the first to cause SARS, that is, severe acute respiratory syndrome, in humans. In November 2002, a previously unknown coronavirus, which came to be called SARS-CoV, was identified in a province in China. It caused pneumonia, and by the end of the outbreak in June 2003, 774 people had died. Public health efforts managed to contain the spread of infection through the traditionally effective methods of contact tracing and quarantine. There was no vaccine, and at the time, no demand for one. It was unclear if or when this coronavirus would return. Coronaviruses are often found in bats, and most usually stay in bat populations, never reaching humans. With SARS, bats likely transmitted the virus to other mammals more commonly exposed to humans. That's also what happened with the outbreak of Middle East respiratory syndrome, also known as MERS-CoV, in 2012. MERS was detected in camel populations in the Middle East and spread to humans in close proximity to them by way of vapor droplets sent into the air when the camels breathed, sneezed, or coughed. With each new outbreak, scientists and public health organizations learned more about which responses were most effective in containing viruses and which needed improvement.
Blink 2 of 7.
With the 2014 Ebola outbreak, it was clear that the systems in place to combat rapidly spreading viruses were inadequate. Ebola outbreaks actually date back to 1976, but by 2014, there were still no licensed vaccines or approved treatments for the disease, despite its fatality rate of between 40 and 50%. Many Ebola vaccines had been in development, but only 2 had been tested on rhesus macaque monkeys, the last step before human trials. By April 2015, the Ebola virus had already spread through West Africa, but case numbers had started falling, so there were fewer areas of high infection available for real-world trials. And that meant scientists could only test one vaccine at a time.
This is the key message. Work on the Ebola vaccine paved the way for enhanced vaccine technology.
Working with the labs, public health organizations began testing one Ebola vaccine known as VSV in one region. The plan was then to switch to the second one in development, called the ChAd3 Ebos vaccine later on. By June 2016, with the help of contact tracing, quarantine and the VSV vaccine, the outbreak had been contained. The ChAd3 vaccine was no longer needed, and there was no demand for continuing clinical trials. When the WHO, the World Health Organization, put out a list of high-priority diseases in 2016, it included SARS, MERS and Ebola, aiming to better prepare for the next big outbreak. It was a perfect opportunity for one of the co-authors, Sarah, and her lab at the University of Oxford to ramp up work on similar vaccine technology called ChAd-Ox1, which they'd been working on parallel to the scrapped Ebola project. Like the ChAd3 Ebola vaccine, ChAd-Ox1 is based on an adenovirus, a type of virus that causes cold symptoms like runny noses and sore throats in humans. Most humans already have antibodies against common adenoviruses. That's why those from chimpanzees, known as simian adenoviruses, generate stronger immune responses in humans. The Oxford scientists took the adenovirus and removed the gene that enables it to make copies of itself and spread throughout a host's body. As a result, it became replication deficient. The gene was then replaced or recombined with a different gene that tells infected cells to produce the virus's protein. This triggers an immune response that creates antibodies to suppress the virus before the recipient ever actually comes into contact with it. In other words, the body learns to fight the virus without ever getting sick. And there, Sarah and her team had it. The basis for a replication-deficient, recombinant simian adenoviral vectored vaccine. By refining this method, they were able to create a new type of platform technology, which is a pre-designed framework upon which vaccines can be built. This would allow them to create vaccines for other diseases quickly, including ones that didn't yet exist.
Blink 3 of 7.
To understand how platform technology works and why it was so useful in accelerating development of the COVID-19 vaccine, consider the analogy of a baker who offers birthday cakes with personalized messages. Let's say you want to order a cake for a friend's birthday. You tell the baker what the cake should say and how it should be decorated. Now, if business is slow, the baker starts the entire baking and decorating process from scratch. But once orders start piling up, that method takes too long. So instead, she bakes and ices many cakes each day, then adds customized messages and finishing touches for specific orders as they come in. By completing the most time-consuming steps in advance, the baker can plug in the missing parts and fill orders much more rapidly. With the ChAd-Ox1 platform, Professor Gilbert and her team were able to use this rapid method to, in a sense, bake the cake in advance, first making an influenza vaccine, then a MERS vaccine, and then moving on to other viruses.
The key message here? The ChAd-Ox1 MERS vaccine served as a blueprint for the SARS-CoV-2 vaccine.
By using the same platform for several vaccine development projects, Gilbert and her co-author Dr. Green at Oxford's Clinical Biomanufacturing Facility, or CBF, were also able to build on their previous research. When the WHO added the unknown Disease X to their list of priority diseases in 2018, Sarah and Kath were already laying the groundwork for the vaccine. Disease X was the nickname of a hypothetical future virus. Scientists didn't know what it would be. But in late 2019, SARS-CoV-2 was identified in humans. As it spread, it became clear that this was Disease X. By the time they'd created the MERS vaccine, they already knew from its 2 successful clinical trials that it was both safe and effective even for children, the elderly, and individuals with pre-existing conditions like diabetes. Since MERS is a coronavirus too, it also contains a type of protein called a spike protein. All they needed to continue metaphorically decorating the new COVID vaccine was this spike protein's genetic sequence. And scientists in China made that available online on January the 10th, 2020. For the vaccine, Oxford vaccinologists just had to take that new genetic code and alter it. The vaccine would then make the body's response more efficient, enabling it to produce the same protein sequence without any of the virus's negative effects. Less than 2 days after the novel coronavirus's genetic sequence was made public, they had already designed the exact DNA sequence for the vaccine.
Blink 4 of 7.
By the end of January 2020, Sarah, Kath, and their respective teams at Oxford were already hard at work making the vaccine and applying for funding. Because the labs had limited capacity and COVID-19 took priority, they also had to put other vaccine development projects on the back burner. The process of decorating the cake, adding the virus's unique genetic material to the ChAdOx1 platform, actually involves several different steps. Normally, each of these steps would take months or even years, and the funding applications alone require a lot of waiting between steps. With this new rapid method of development, however, Sarah and Kath's teams at the clinical biomanufacturing facility could move on to the next step before all the testing was finished on the previous step, an approach known as proceeding at risk.
Here's the key message. Progressing from stage to stage at risk greatly reduced the timeline to make the COVID-19 vaccine.
When scientists talk about proceeding at risk, the word risk doesn't refer to the safety or efficacy of the vaccine or the safety of those developing it. Rather, the risk is to researchers' time. And for labs which rely on external funding, time means money. Here's the downside of this approach and why it can be so risky. If testing finishes and is unsuccessful, all the work on the stages that were to follow has been done in vain. Scientists then have to return to the earlier step and get it right before moving on again. And yet, in the case of the swiftly evolving COVID-19 pandemic, the potential benefit far outweighed the risk. If tests at every stage were successful, millions more lives could be saved. It was a risk they decided to take, and it paid off. The first steps of development went as planned, and cells that had been infected with the modified SARS-CoV-2 virus were ready to be harvested and purified. To remove the parts of the cells the researchers didn't want, the cells were placed in a centrifuge and spun for 2 hours at 154,000 Gs. For comparison, the fastest roller coaster produces a G-force of just 6.3. Finally, on April 2nd, 2020, CBF staff began filling vials with the purified vaccine, labelling them and testing each vial to make sure it was sterile. From the time they designed the vaccine in January to the end of labelling and testing the vials, a mere 65 days had elapsed. Vaccine production was now ready to scale up.
Blink 5 of 7.
The number of lives a vaccine can save doesn't just depend on how effective it is in trials, but also on how many doses you can produce, how many people will have access to it, and how many will choose to get vaccinated. In order for the latter two factors to even come into play, the scientists at the CBF in Oxford needed to produce their vaccine at a much larger scale. At the CBF, they could grow 10 litres of vaccine culture at a time, yielding several hundred doses. At Advent, their partner lab in Italy, the capacity was 100 litres of culture, meaning thousands of doses. This was enough for small trials, but what they actually needed was the capacity to produce millions of doses at a time.
The key message? Partnering with AstraZeneca and others provided the funding and infrastructure to manufacture hundreds of millions of vaccine doses.
One member of the Oxford team, Dr. Sandy Douglas, had considered the problem of scaling up production from the early stages. He'd developed a method to use 1,000-litre bioreactors, basically enormous tanks, to grow and purify millions of doses at once. They had already tested this technique at a smaller scale, so Dr. Douglas applied for financial backing to prove its true potential. With £400,000 in funding, he successfully tested out his large-scale manufacturing method with 50 and 200-litre tanks at the Poole facility in Portsmouth, UK. He then secured more funding from the UK government's vaccine task force. This allowed his collaborators to optimise the production method, as well as test, store and transport the doses produced. By the end of April 2020, everything was set. The CBF was producing enough doses for the 3 phases of human trials, and the systems were in place to produce millions more once the vaccine was approved for the general public. On April the 30th, it was announced that the Institute would be partnering with a large pharmaceutical firm called AstraZeneca for global production and distribution. Orders for billions of doses of the vaccine, which had been renamed AZD1222, began pouring in from around the world. The Oxford-AstraZeneca partnership led to another key feature which would set their vaccine apart from most of the other COVID-19 vaccines. It didn't need to be stored and transported at ultra-cold temperatures. Since it could be refrigerated normally, between 2 and 8 degrees Celsius, like the vaccines for polio and measles, it would be that much more accessible to parts of the world that didn't possess or couldn't afford such facilities.
Link 6 of 7.
While designing and producing the first COVID-19 vaccines took just over 2 months, it was all in preparation for the final stage before licensing: efficacy trials. This stage would take a further 7 months. Luckily, the same ChAd-OX1 platform was employed in previous influenza and MERS vaccines, so much of the early testing on dosing had already been completed. But since this was a brand new virus, and very little was still known about its behaviour, scientists at the CBF and the University of Oxford's Centre for Clinical Vaccinology and Tropical Medicine, or CCVTM, decided to be extra careful. Certain vaccines have the potential to cause a worse infection in recipients if they come into contact with the actual virus later on.
Here's the key message. Rigorous testing ensured the vaccine's safety and efficacy before its public release.
Exercising caution meant finishing animal trials before giving a single dose of the vaccine to humans. To do this, scientists vaccinated animals and then exposed them to high concentrations of the coronavirus. It was a success. The vaccine generated the correct immune response. The vaccinated animals also fared much better than the control group of unvaccinated animals. With this news, the teams back in Oxford got the green light to start organising human trials. The first round of tests, called Phase 1 trials, started with just 1,000 volunteers. The goal was just to see if the vaccine induced a safe immune response in healthy adults under the age of 55. Half of the volunteers received the vaccine, while the other half got a placebo. As a double-blind study, neither the volunteers nor the nurses administering the vaccines knew if they had the real vaccine or just a placebo. All volunteers kept health diaries online, documenting any health issues that arose. 4 weeks later, a group of 10 volunteers were given a second booster shot to see if it improved their immune response. For the Phase 2 trials, the age range of volunteers expanded to include adults aged 56 and up without pre-existing conditions. Finally, Phase 3 was open to adults of any age and any health condition. The trials showed that receiving 2 doses 12 weeks apart produced the highest efficacy levels and reduced infection, transmission, and symptoms in cases of exposure. The trials were then expanded to Brazil and South Africa, and on December the 30th, 2020, the UK's Medicines and Healthcare Products Regulatory Agency, or MHRA, approved the AZD1222 vaccine. The first dose was administered on January the 4th, 2021, with hundreds of millions to follow.
Blink 7 of 7.
With the first vaccinations being injected into arms around the world, the work of reining in the virus and preparing for the next one was just getting started. Along with the logistical hurdles of production and distribution, there was a more human obstacle. Global vaccination campaigns have, in certain communities, been met with skepticism, posing a threat to the vaccine's overall effectiveness. There's also the issue of variants. The more a virus spreads, the more it mutates. The vaccine has been shown to be effective against some variants, but for others, modified vaccines will be required. While certain preparations accelerated vaccine development and the global effort to save lives, there is a long way to go in optimizing the processes for the next viral outbreak.
The key message here is, to prepare for disease Y, the world needs to improve its research and manufacturing infrastructure, public health response systems, and global cooperation.
Epidemiologists already have some indications of what this future hypothetical virus, for the moment nicknamed Disease Y, might look like. One clue lies in industrial farming practices, which often keep animals in unsanitary conditions. Such environments lead to viruses being transmitted from one species to another, mutating, and eventually reaching humans. Disease Y could be another coronavirus, like SARS or MERS, or even a new highly infectious strain of the influenza virus. Or it could be a completely unknown virus. Scientists have still only studied 267 of the estimated 1.67 million viruses in existence. Disease Y, whatever it turns out to be, will test the world's response systems once again. One big hurdle during the COVID-19 pandemic was funding and manufacturing infrastructure. Early on, the CBF applied for a few £1 million in funding. At the time, it was seen as entirely unreasonable. But in hindsight, it's just a tiny amount of the money governments and companies have spent to combat this virus. Organisational bodies also need to ramp up programmes that stockpile important equipment like PPE for labs and healthcare workers. Lastly, investing in pandemic preparedness means improving upon the vaccine development methods pioneered by the CBF and others. If they can speed up the development process even more, and if other vaccines such as Pfizer, BioNTech and Moderna can be modified to be refrigerated at normal temperatures, the overall pandemic response will be that much more effective the next time around.
You've just listened to our blinks to Vaxxers by Professor Sarah Gilbert and Dr. Catherine Green.
The key message in these blinks is that much of the world was unprepared for the challenges brought about by the COVID-19 pandemic. Scientists at the University of Oxford, however, were among those preparing for such an outbreak long before it occurred. By developing vaccines against other coronaviruses like SARS and MERS, as well as Ebola, they built the research basis necessary to create the ChAd-OX1 framework, which produces vaccines much more rapidly than normal. After designing, manufacturing, testing and distributing the COVID-19 vaccine, it's important to look toward the next pandemic. By improving the infrastructure for vaccine development and global cooperation, the world will be better prepared to tackle future outbreaks.